Okay, we're going to change speakers now to the second speaker in this session, Dr. Laila Abdullah, and then we'll have a question and answer period afterwards. So Dr. Laila Abdullah is a scientist, senior scientist at the Roskamp Institute. She's a neuroscientist with a history of working in biotech and translational scientist who moves things from the lab through animal work into human trials. And it's been exciting to work with her in the Gulf wilderness area for the last, I don't know, more than a decade. Dr. Abdullah is skilled in molecular biology, applications of lipidomics, metabolomics, proteomic-based mass spec technology. She's a leader in designing and implementing randomized double-blind placebo-controlled clinical trials based on very strong basic science. And she also works with the Roskamp with their doctoral program in partnership with the Open University. Laila, take it away.
Well, thank you for the invitation and the opportunity to speak to our veterans about our work. And thank you for highlighting all the areas that I focus on. But for the purposes of this talk, I will be focusing on some of the work we're doing in relation to the immune responses. So I'll be focusing on that today. Just to kind of remind ourselves, why focus on the immune aspect? Number one thing is that of all the systems in our body, immune cells and immune system is capable of remembering what happened to you a long time ago. I mean, that's why we have vaccines, and that's why we have, responses to viruses. So focusing on and remembering that important aspect of the immune system, we focused on the two branches of the immune system. Firstly, just in terms of the prior evidence why immune system is overcharged in veterans with GWI. We've seen reports of circulating high number of IgGs and IgM and changes in blood lymphocytes, and some of the pioneering work that was done in the area by Nancy, by your team, and we took a lot of lessons from that to move forward our own work in this area. Changes in inflammatory cytokines. We wanted to focus on how a chemical exposure can affect the immune system. So we started to look in this area a few years back, and what we were finding is that the reports of the certain chemicals and the drug breakdown products that can then have the capability to combine with our own body's proteins. And so in a way that when they combine, they look to the immune system, particularly the cells that patrol and look for, foreign antigens. They mistakenly recognize these combinations as foreign and mount an immune response. So kind of working under those hypotheses, and maybe certain chemicals and certain pesticides may have triggered that kind of immune response. So we focused on that for this work. And we also focused on, what happens at the cellular level. So just a reminder for us, there's two branches of the immune system. There's the humoral immune responses, which involves B cell and antigen production, which basically, this the patrolling mechanism, and then the cell mediate, which is more targeted approach that our body and our immune system takes to eliminate threat. And so, focusing on those two different arms, what we previously showed and published is that in our mouse model, we found that a metabolite of permethrin is called 3-PBA. We previously found that it, and by mass spec, we looked to see whether these combinations were present in our animal models, and we could see that months later we could see these combinations in the blood and in the brain of the mouse models. And then what we thought, okay, is our body reacting to it? So we looked for antibodies, and again in our mouse model we saw that certain combination of these chemical breakdown products with our protein. In that case, we used albumin. We saw that these combinations can be recognized by the immune system and was seen to be altered in in our mouse models and some blood samples from the veterans with Gulf War illness. So that was some of the initial work that went into this, and we followed up. We're now focusing on organophosphate, particularly chlorpyrifos, as in fact that it was used during the war, Gulf War, and the spraying the tents, and for a long time, even after in post 9/11 era, it was still being used to spray the tents and as fogging agent. So we're focusing on that right now. And then we also wanted to look to see in the blood cells from veterans relative to healthy Gulf War vets, is there a change in how these T cells, the cells that coordinate targeted response, how they respond to stimulus, and some of that focuses on generally Gulf War veterans fine, and then for example we get some veterans come through and then they'll go by our lab and they'll be like, oh, I can't go in there, and then it will trigger certain responses. So we were kind of trying to get to understand that and focusing on the immune system. So the first thing here, as I mentioned, we're focusing on chlorpyrifos. What we did is in our mouse model, we saw that chlorpyrifos does in fact, when it's in the body in our mouse models, it does attach to certain proteins. We found that it can enter the brain and form addicts. What we call addicts is just a combination of chlorpyrifos with certain proteins that it forms linkage with proteins. We saw that in the brain of our mouse models. And again, in the blood, the most prominent protein is albumin, and we saw that as well. So here, what we did is we combined chlorpyrifos itself. We combined it chemically with albumin. We developed an ELISA system where we coated the ELISA plates with combination of this protein and chlorpyrifos. And we then tested antibodies from healthy Gulf War vets. Basically, we applied plasma to it, and then we checked to see if there are any antibodies that recognized that combination of chlorpyrifos with albumin. And just keep in mind the way this is, it's an ELISA-based system, so whatever antibodies are in these plasma samples or in our blood samples, they're recognizing the shape that is on these ELISA system. So, that I'll bring that up later, and that comes to the specificity of those antibodies. But all we can say is that the antibodies that we detected, they're recognizing the shape that resembles a combination of chlorpyrifos and albumin. So we, as you can see on the table in the top here, we've got 41 healthy Gulf War veteran controls and about 110 Gulf War veterans. Gulf War veterans were slightly younger than the control subjects. So, and then they were similar on age and other demographic factors. What we can see here is that we divided the antibodies in quartiles. So, basically, we looked at the highest quartile here in combination. So you can see that only 12% of the control veterans were experiencing or were showing these antibodies, compared to 28% had the highest quartile. Here is a description of all the antibodies that were detected, and you can see that veterans with GWI seem to have higher presence of these antibodies in their blood compared to healthy Gulf War veterans. So, as I said, we have these Gulf War veterans, and we're probably us. Chlorpyrifos is pretty abundant. So it's recognizing the shape. Question about specificity still outstanding. We're working towards that. Some of our hypotheses involve, after this initial exposure, are these cells going around looking at other proteins in our body and looking at shapes that match those and then attacking that. That's part of the autoimmune phenomena that occurs. So we're looking into the mechanistic aspects of that, and that's part of our ongoing work in that area. So the next thing that we focused on is understanding how these T cells respond to certain stimulus. So we set up a bead-based system, which is designed to activate the T cell receptors. It allows us to expand the T cells that we've collected from our veterans' blood and expand it up so we have enough cells. Then we look to see how they're responding to this stimulatory signal and whether they are behaving in a fashion that promotes inflammation, or are they behaving in a fashion that dampen down the immune responses. So we looked at, these are the cytokines that are involved. We looked so this is we basically took the ratio of baseline, what the baseline characteristics were of these cytokines, and then what did they do after the immune responses. So you can see that, of course, the healthy Gulf War veterans cells increase their TNF alpha, but as you can see, that veterans with GWI really significantly increase TNF alpha, which is a very potent pro-inflammatory cytokine. We also saw that IL-6, which is also an inflammatory cytokine increase, and IF and gamma. So a number of these cytokines that are involved in inflammation, they tremendously increase their levels compared to control. And so their response seemed to be very much heightened. So it's like they're ready to charge, and then as soon as they see a signal that stimulates the T cell receptors, they rev up, and then they start responding. So it's a heightened response that's beyond normal. What was also interesting is that these cells increased IL-4 levels as well, which is generally considered an anti-inflammatory cytokine. And I think, Nancy, some of your earlier work pointed to a mixed Th1, Th2 response, which I believe is what we're seeing here, that these immune cells indistinguishably just go on an attack mode, increasing both pro and anti-inflammatory responses. And to better understand that is the next goal of the study. So in conclusion, we basically are focusing on understanding how these two branches of the immune system are behaving in veterans with Gulf War illness, and what their collective output looks like. We're understanding whether or not, what is the specificity of these antibodies, how specific they are. There's a lot of work to be done in this area, and can they ever be useful in diagnosis? And we're trying to understand these mixed Th1, Th2 responses, and how do we target that therapeutically, which again presents a challenge. So I will thank, of course, all the Gulf War vets who have taken part in the study. As everybody's mentioned here, this work would not be possible without their support and without them taking part in the study. And of course, the B-Brain, a great resource, allowing us to have access to samples that would have taken us years to complete. We are still recruiting for these studies, and we need your help. So there's a number here I'm including. And of course, the VA and the CDMRP for funding this work. And yeah, I'm happy to take questions.